Hello everyone, I'm here to talk to you about thermodynamics. We're talking biochemistry here. Let's get into it. We're going to be talking about thermodynamics from a biological perspective. Of course, you can do thermodynamics and thermodynamic equations in just regular chemistry, general chemistry, organic chemistry, but we're dealing with biochemistry here, so we're going to be talking more about the biochemical side of things. This equation, it looks complicated. It's not. I'm not going to say it's simple, but the concepts are actually really important. And so let's get into it. So we're talking about the free energy of a system. So basically the free energy is like the energy available to do stuff. Okay? And so when we deal with that in biological systems, what we're really looking at is bonds. Are we going to form bonds? Are we going to break bonds? What do we do with the energy? If we want to have something happen, do I need to break some other bonds to make that happen? Can I break the bonds in that molecule? This is going to help us do that, okay? And so the key point is that thermodynamics, at least in biological systems, tells us if a process is going to happen spontaneously or not. And that's a really important word, spontaneous. I want to talk about that. And it's going to be based on the enthalpy, it's always a hard word to say, entropy, and the temperature of the system. So here we have our enthalpy or enthalpy. We have our entropy, which is delta S. The delta means change in, right? So products minus reactants. So we've got our delta H, we've got our delta S, and then we have our temperature of the system. And those together form our delta G, which is sometimes called Gibbs free energy after a dude named Gibbs. But it's just the free energy of the system. And that's kind of taking all these variables that can occur and then using those to help us just make a decision, basically. Okay, so we're going to combine a bunch of stuff to make delta G. And we're going to talk about spontaneity. Okay, now just because something, this is an important distinction, just because something is a spontaneous reaction, okay? So if our delta G tells us that the reaction will be spontaneous, that doesn't mean instantaneous. And a lot of people mix those things up, okay? Really, really important distinction. Just because it's spontaneous means it's energetically favorable between the products and reactants. That doesn't mean it's just going to happen, okay? Because oftentimes, actually most of the time, we need to put a little bit of extra energy into the system. We're looking at the difference between our product and reactant energy, not whether or not they will just spontaneously occur, or because most of the people think that just means it happens like immediately. Oh, it just happened because it's delta G said it would happen. No, it could take years, it could take decades. If you don't give it that little boost to get over that hump of the energy, which I'm going to show you some graphs to talk about this. But anyway, so instantaneous versus spontaneous, big difference, okay? But we're going to talk about spontaneity here. So let's get into it. I want to talk about the subdivisions of this equation, okay? So enthalpy is the first thing we're going to talk about. And I realize if you've taken organic chemistry or maybe even gen chemistry, you probably have talked about this before, but I'm going to assume that if you have learned it before, that it's going to be need a little refresher. I always need a refresher, so let's talk about it. So enthalpy is basically the energy contained within bonds, okay? So when we have our delta H, that's the bond energy of our products minus the bond energy of the reactants, okay? So if your reactants is really, really big and your product is small, you'll end up with a delta H that's negative, right? And so that means that heat is released. So if you see a negative delta H, that means heat came out of the system. We call that an exothermic reaction, okay? So if something is exo, meaning out, thermic, meaning heat, so heat was released, i.e. bonds were broken. This is what you're usually going to think of. This is the first thing I think of when I think of like a reaction. Oh, I took like a glucose molecule and I broke the bonds and I pulled the energy out. The delta H was negative. It had a negative enthalpy. It was an exothermic process. Heat was released. Now what we do with that heat, I'm using heat a little generally, what we do with that energy that comes out, well, that's a better question, and that's what we're going to talk about later. Now, if your delta H is positive, this happens also all the time. The heat was absorbed, so we made bonds. Okay, we call that an endothermic reaction. Okay, the heat was absorbed by whatever we're making, and so in this case, if it's endothermic, that would be, let's say, for example, what plants do. So plants are going to photosynthesize. They're going to use the energy from the sun. They're absorbing that heat. and they're going to use it to take carbon dioxide and water and make glucose molecules. That's an endothermic reaction. Okay? So that means we're forming bonds. And so you can generally look at a reaction by itself and say, is it exothermic or endothermic? Did I break bonds or did I make bonds? Okay, so that is your enthalpy. And here is a great example. We're going to talk about ATP a lot. ATP, adenosine triphosphate, and breaking ATP into adenosine diphosphate with an inorganic phosphate group. That is kind of the key molecule for all of the thermodynamics that occur inside of biochemical reactions. So in this case, thank you Khan Academy for making a really nice diagram here. We're going to follow ATP. So we've got our little ATP. We talk about it a lot. It's like our smallest form of a battery. Okay, we're going to use the electron transport chain at the end of glycolysis and the citric acid cycle to make a ton of ATP. And that's going to be our little battery that we're then going to couple with other reactions. And so when I do that, when I break this phosphate off, so I've got 3 phosphates here attached to the adenosine, when I break that phosphate off, that is breaking a bond, right? That is an exothermic reaction. I'm releasing energy. It's a negative delta H. So if I asked a question, negative delta H, there you go. That's an exothermic reaction. Pretty straightforward. Now, if I have my ADP and inorganic phosphate and I want to make it back into an ATP, well, I have to put a little bit of energy in, and that's where we get glycolysis and the citric acid cycle, putting those hydrogens on the other side of the electron transport chain and letting those pass through, which that's way later in biochemistry, but we'll get there. When I mean to make those bonds, I'm building a bond, right? I'm taking 2 things and putting a bond, reattaching a bond. That is an endothermic reaction. It has a positive delta H. Try and avoid making any positive or negative associations with either one. Sometimes you can say like, oh, an exothermic reaction, like that's a good thing. It's making heat, I'm breaking stuff. Whereas endothermic is kind of a bad reaction. I don't know. That's just kind of what was stuck in my head for a long time. Try not to put emotions into your reactions, okay? So that is enthalpy. So let's get to entropy, which was always my favorite thing. So when we have our chaos or order of our systems, that's something that we can actually measure. It's usually pretty small. Okay? It's basically a measure of how energy is distributed in a system. And so you can measure it with a calorimeter, but basically what we're trying to say is chaos. So we're measuring chaos. If things get more chaotic, okay, that means you have a positive delta S. So delta S is the change in chaos. So S is chaos. So if my chaos is going up, it's bigger, it's positive, okay? That means I'm becoming more chaotic. If it's negative, that means overall I'm losing chaos and thus becoming more ordered. And so when you see entropy referred to, sometimes we'll talk about it. I like to refer to it in terms of how chaotic is the system, but other people like to look at it the opposite way and say, okay, how much order is there in the system? And it just kind of depends on how you want to view it. So if your delta S, right, your chaos is negative, that means I've lost some chaos. Okay, I have gained order. So a super common example, I just picked this because it's like the most 2002 picture I've ever seen of like a dorm room. I love these computers and the TV before LCD screens. So if you have a nice clean dorm room, it took some energy, right? Okay, so you have a negative delta S for your dorm room. Okay, it's cleaner. I apologize for the quality too. Like I like this concept because I don't know, you're college students. So if your room is nice and ordered, it has negative delta S at least over time. But if it becomes more chaotic over time, that's a positive delta S. Okay. Remember that we're always flowing towards chaos. That is the second law of thermodynamics. The trick is with biological systems. And this confuses a lot of people. When we look at biological systems, yes, the entire universe is heading towards chaos, but biological systems can temporarily have order within them, okay? And so that's what I'm kind of showing here. When we have our energy, biological systems can pull in extra energy either through sunlight or by eating other biological organisms. We take that energy in and basically we use ATP to build new molecules to go against the chaos, okay? But in the end, we will all lose to the second law of thermodynamics. All that heat and all that waste will eventually head out, and we will head to our lowest energy state, the highest entropy state. Okay? So, not to terrify you, but it is a little existentially terrifying. So let's look back at our little Khan Academy picture here. So entropy, I love it because it's really straightforward. How many things do you have? If you got one thing going to two, well, that seems more chaotic to me. If I have an apple and I cut it in half, now I have two pieces of an apple, okay? I can throw a piece of apple at two different people. If I only have one apple, I can only throw one. I have an apple on my desk right now. That's why I'm making that analogy. So that is more chaotic. So that would be a positive delta S. I have increased the chaos of the system. I have two things I can throw at people. Now, if I take my ATP, my ADP and my phosphate and smash them together and build a bond, then I am now less chaotic, right? I took two things and I became one. So that is now less chaotic. So that is a negative delta S. There's less chaos in the system. Cool. Awesome. I'm kind of ignoring temperature for now. Temperature of the system is usually relatively constant. And so as temperature goes up, it can affect, or down, it can affect chaos, right? So that's why the temperature of a system is intrinsically tied to the chaos of the system, which makes a lot of sense. Temperature, really in a nutshell, is the vibration of atoms, right? So as temperature goes up, the chaos is gonna go up, okay? And those are really, really closely tied. And so temperature can really throw a curveball at your reactions of whether they're spontaneous or not. And temperature a little bit too. So coming back to Gibbs free energy. So the free energy of the system, our delta G. So what that's gonna tell us is spontaneity, okay? So usually the only term that we're really, really worried about is the delta H, okay? Are we forming a bond or are we destroying a bond? That's gonna be the biggest impact on what your delta G is. They're almost equal, but temperature and chaos can bring a little bit into that. So if I have a huge positive delta H, so if I back up a little bit, when I have a positive delta H, that's when I'm building bonds, right? So if I have a really big positive delta H, it's going to take a lot of, my chaos would have to be huge, my temperature would have to be huge to actually pull that out, to basically eliminate or cancel out the delta H, okay? Now if I have a big negative delta H, any temperature that goes up and any delta S that goes up, and these amplify each other, if this is huge and this is massively negative, then I'm going to have a huge negative delta G as well, okay? So just if you look at the equation and just kind of look at it, and what I like to do, what I used to do is just put random numbers in there. So like just put like a really big number into delta H and normal numbers into T and delta S and see what happens, and then just kind of switch things up. But basically, in lieu of doing that, I just want to talk about if your delta G is negative, it's a spontaneous reaction. Now, it is not an instantaneous reaction, that's what I was talking about, but between your products and your reactants, it is spontaneous. They want that to happen, okay? Now, we can go against that, and we'll talk about that in a second. If your delta G is positive, it's not a spontaneous reaction. I also want you to avoid applying any kind of emotional connection to that. Some reactions are spontaneous, some are non-spontaneous. It's just how things work. You're gonna see this a lot, and I wanted to mention this. On the MCAT, they will ask you at least the basic concepts behind this. They're not generally going to just give you these equations, because there are deeper equations involved with this other than this one. But the concepts that are going on of an exothermic reaction and an endothermic reaction, they will ask you those kinds of questions. Quickly, we're going to talk about this table a little bit. We're not going to spend too much time on this. But what I want you to get out of this is that the delta H is your key into what's really going to happen. Is it going to be spontaneous or not? So just look here at an exothermic reaction. Okay, so think breaking ATP into ADP and a phosphate, which is how we're going to get a lot of energy into our biological systems. That wants to happen. Okay, at the temperature, as long as we're increasing the entropy, how in the world would you decrease the entropy? The odds of that happening, it doesn't really make sense. And so you're usually going to be spontaneous because you're taking one thing, cutting it in half, and making two things. That's going to cause an increase in entropy. It's just basically going to be spontaneous at any temperature. Now, the opposite is also true. So if we're going to build bonds, okay, we're going to usually see a decrease in entropy. Okay, so it's going to be non-spontaneous at any temperature. Okay, so if we're building bonds, of course, we're going to usually decrease in entropy. Other two possibilities, there are reactions that can be endothermic, so we're building bonds, but can cause an increase in entropy. But usually what we're going to see there is that, remember that temperature in our equation is intrinsically tied to our chaos, okay? And so if our temperature gets really, really, really big, it amplifies our delta S, okay? And so that can kind of overcome the power of the delta H. So delta H is usually what we're referring to unless temperature is really changing things here, okay? So in this case, if I have a really high temperature, okay, so really big temperature is going to drive delta S to be really huge, and it's negative T delta S, right? So this multiplier is massive, which makes this whole thing massive, which makes this giant whole chunk of this a giant negative number, and it can overwhelm a positive delta H. So if I have a positive delta, there can be reactions that occur in these areas, and they all depend on temperature. So remember that temperature is a multiplier of the chaos. So if you can drive your temperature really high, you can amplify the chaos. And so even if you were having an endothermic reaction, so you're building bonds, you can actually increase the entropy if the temperature is high enough. And so that would be spontaneous at a high enough temperature, because that temperature, heat is energy, right? And if you increase temperature, that means your heat is going up. That means there's a more available energy. So if I pump enough heat into the system, yeah, it's going to cause chaos, basically. And the opposite is also true. If I pull all the heat out, I can actually take an exothermic reaction and cause it to not happen. Anyway, so let's keep going. I want to get into biological systems and talk about this a little bit more. Okay, let's get to some more practical stuff with biochemistry. Biological reactions can only really go two ways. Are you going to break things and get the energy and the byproducts out, or are you going to use energy and build new things? Okay, our temperature is going to stay constant. We need to know if the reaction is going to happen or not. That's where our delta G is going to come in. And ATP is going to be our key into these reactions and what actually is happening, okay? So a term that I did not mention yet, I talked about exothermic and endothermic reactions. That's if you're referring to enthalpy. If you are referring to delta G, which we usually will be, you're going to see these two other terms get thrown around. Exergonic, so it's got that G sound in there to make you think it's a G. So exergonic and endergonic. They match up with exothermic and endothermic as well. Okay, so basically we need to just talk about ATP as a reaction, okay? Breaking ATP into ADP and pulling out that energy, okay? So if I have a negative delta G, that's when I'm taking ATP and breaking it and getting that energy out, and I can do stuff with that energy. That's an exergonic reaction. If I'm going to build my ATP back up using the electron transport chain, then that is an endergonic, okay? So I'm taking energy from somewhere else and putting it into the system to build this bond right here, to take this phosphate and attach it there, okay? The energy actually comes from putting hydrogens on the other side of a membrane and creating a concentration gradient. That's where that power comes from. Okay, so two terms that you need to know. All right, let's keep going. I just want to talk a little bit and get you exposed a little bit to where the delta G's are coming from. Because from now on, we're not usually going to mention delta HT or delta S. We're going to just stick with our Gibbs free energy, okay? So let's look at where the Gibbs free energy for ATP comes from, okay? Because we're going to see it all the time. So we need to talk about that. So we're going to take ATP, okay, and we're going to break it with water. We're going to do some hydrolysis. And if you think about that, so we're going to take two things, ATP, adenosine triphosphate, and a water molecule, so that's two things. And we're going to break it into three things. We're going to take it, break it from ATP into ADP plus a phosphate plus the products of water too. So like there's like a hydrogen float around. But basically, so we're going to have two things and we're going to make it into three things. Awesome. So our chaos is going to go up. So you would have a positive delta S. It turns out that's actually a really, really small number. Your chaos, think about the chaos of the universe compared to the chaos that's happening of one single reaction. Your chaos is really small, and that's what we see. It's incredibly small. It's 0.0054 kilojoules per Kelvin. Okay, so Kelvin is the temperature scale that we have to use for these equations. It's just what we have to use. And then, okay, so that's really small. And then at standard temperature and pressure, so standard temperature in chemical reactions is 298 Kelvin, which I think is 25 degrees Celsius. And so that's where that comes from. So that's going to multiply by that. Okay, so it's still a really small number because this number is going to pull this guy way down and make it small. And so our negative number is really small, right? And then we're going to come over here, and there's our delta H. Okay, and now we're breaking a bond, right? So I'm taking ATP, and I'm going to break a bond. So that's an exothermic reaction, and that's going to have a negative delta H. Okay, so I'm releasing energy out of that by breaking that bond. And I'm going to end with a negative 31.6 at standard temperature and pressure. At body temperature, which is where most of our biological reactions are actually going to occur, so all we're doing is we're just amplifying the chaos a little bit. So we've taken some energy, and instead of 25 degrees Celsius, we're jumping up to like 35 degrees Celsius or 37 degrees Celsius. It kind of depends on what you want to look at. Different animals have different temperatures too. And so we're going to just put a little bit of heat energy into the system. And so we should get a little bit more delta G out of this. Remember, negative delta G is energy that we can use, right? So if it's negative 31.6 at standard temperature and pressure, we put a little extra energy in from our temperature, and now we're at negative 31.7, which seems negligible, but that can be a big difference when you get down and zoom in on biological molecules. So that's kind of cool. Now, we're going to see our delta G all the time. Okay, so you're going to see the delta G for ATP a lot. So this 31.7 is going to come up all the time. We see the delta G for lots of other reactions as well. So here are some common ones that actually result in a negative delta G. We're going to see a ton of positive delta Gs as well. So those are reactions that are non-spontaneous, right? That doesn't mean we can't get them to happen. It just means we need to use a little extra energy to get them to happen. Okay, they are by themselves non-spontaneous, but we can put extra energy in and get them over that hump, okay? And we're going to talk a little bit about that. But these are some other reactions that you can use that have a negative delta G, and you can use these reactions outside of ATP going to ADP to actually drive reactions. So going from ADP to AMP is actually another big one that you can use to do a lot of stuff. So I don't want you to think that ATP going to ADP is the only way that we can get a negative delta G, but that is the most common one, and that's the one we're going to focus on, especially when we talk about glycolysis, because that's going to be a huge chunk of this biochemistry course, is talking about glycolysis. How do we actually take a large glucose molecule and break it down, okay? So we have to put some energy in to do that. Speaking of glycolysis, I want to show you some of the intermediates in the glycolysis pathway. Not all of these are, but some of these are. And so I just want to show you if we're actually going to break things. So if in glycolysis, we're going to take a glucose molecule and we're going to break all those bonds, and we're going to end up with some carbon dioxide, we're going to end up with a little bit of water and all the energy that came out of breaking all those bonds. Now, it's going to take a little bit of energy to get us over the hump to actually get that happening. But in the end, we can get a bunch of energy out of that. Okay, and that's where I want you to see how many, it kind of depends on which side of the Atlantic you're on, but whether you use kilojoules or kilocalories. But suffice to say, you can get a ton of energy, a ton of free energy out of these systems once you get the ball rolling. And we're going to use enzymes to do that. That's a different lecture. But I just did want to talk about all the energy that we can get out. Notice that these are all negative delta Gs, okay? And just to kind of give you a visual, it's not the greatest quality, but it's just an older diagram, but I like this a lot, just to kind of show these are all the intermediates of glycolysis. And you can see we're still getting a negative delta G, so we're still getting energy out every time we go between an intermediate, but the amount actually goes down until we actually get all the way to the end, and then we're left with these products that we need to get rid of, okay? And so that's just the delta G of glycolysis. It's going to come up a lot. When we actually talk about glycolysis, you're going to see delta G all the time. Moving on to a little bit more practicality as far as our delta Gs are concerned, we want to talk a little bit about catabolic and anabolic reactions, okay? Because we're going to encounter both of those in our biological systems. And so these are things that you may have heard of before. So catabolism versus anabolism. You think of like anabolic steroids or your metabolism. Okay, so your metabolism it gets thrown around by nutritionists and people like that. Really, we're just trying to balance between catabolic, which is the burning of energy or the releasing of energy, and the consumption of energy or the building of things. Okay, and so on the top here, you can see we've got all the things that we're going to eat. Okay, we got our carbs, our fats, our proteins. You can use nucleic acids as well and eat those. And we're going to take those things and we're going to perform catabolic reactions on them. We're going to cut them up and break them up into smaller chunks, pull the energy out so that we can use that energy to then do other things. So this is why you eat sugar and other things, meat, I don't know, chicken, whatever, and you're going to eat that, you're going to break those things down, and then you're going to use that to build parts of your own body, right? And so that's the anabolic side. That's the side that's going to actually build new things. And so you can see here, these are where we actually get some of these pathways to build new things, right? So you can eat a candy bar, you break it down into basic sugars, and then you can take that glucose and you could use it as a long-term storage unit, like a glycogen or a larger chain starch, which we'll talk about when we actually talk about carbohydrates. You've got to make your RNA, your DNA, your proteins, all kinds of stuff, right? And so that's catabolic versus anabolic. Now, as we're kind of going through here, I want to emphasize the idea of anabolism and catabolism, okay? So if we're going to consume things, right? So this is our catabolic, so we're going to eat stuff, we eat our food, and we're going to break it down, and we're going to get the energy out, and we're going to have waste products over here, and we're going to make ATP, right? So follow my cursor here. So we're going to make some energy, we're going to make some ATP out of that, right? We have an exergonic reaction that's going to make some ATP. So catabolism and exergonic reactions are intrinsically tied. We're going to make some ATP, and that's going to be our bridge, our gap bridger. Okay, and we're going to go over here, we're going to use that to make stuff. And remember that ATP is the key molecule here, okay? And so we're going to have our endergonic reactions over here. So we have to put energy in. Remember, these are non-spontaneous, okay? So we have to put extra energy in to build those bonds and actually make them happen, okay? Just because they're non-spontaneous doesn't mean they won't happen. It just means we have to put extra energy into the system, okay? Let's keep going. So these are our two possibilities. So on the left, we have our exergonic reactions, right? So these are ones that we're going to release energy. And this is where it gets a little tricky in that what we've been referring to with Gibbs free energy is we're comparing the reactants to the products always. But if our free energy of our reactants is greater than that of our products, then the difference is the amount of energy that gets released, right? These are, think of this as like a glucose molecule, and this is the breakdown of the glucose into its byproducts, okay? So we've shattered that glucose into little chunks, okay? Over here, think about like what a plant would do. It's gonna take carbon dioxide and water, and through the magic of biochemistry, is going to build a glucose molecule. And so it's less chaotic, that is true. It's non-spontaneous, we have to add extra energy in. So the energy that is stored within here is different from our reactants to our products. So in an exergonic reaction, we're gonna break bonds to get energy, okay? That's the most basic thing to think about. Negative delta G. If it's endergonic, okay, we're going to build some stuff. We're going to take some energy and put it into the system. Now, this big hump right here, that's the tricky part. So that's our activation energy. I'm going to talk about it a little bit in this lecture, but I will talk about it more when we actually talk about enzymes. So let's dig a little bit deeper into what's going on. How can I get this stuff to happen, okay? Just one little side note. Remember that it costs extra energy to make bonds, and that's why sunlight is great. Plants use sunlight because that's all the extra energy they can pack in here. We will eat plants or other animals that eat plants, and that's where we get the energy to build these things, okay? And we do that a ton. So remember, the whole point of things on this planet is to make babies, okay? Now, you can't make babies if you don't have cells. And you can't have cells if you don't have a phospholipid bilayer. And you can't make a phospholipid bilayer out of phospholipids without fatty acids. So you have to make fatty acids if you want to make babies. See? Perfect logic. One fatty acid costs 1,000 ATP. That's a lot, but it's worth it because I have to make babies. I'm a biological organism. That's my goal. And so I need to spend that ATP, which that seems like a lot, but I don't know. You actually plow through that pretty fast. So how do we get a non-spontaneous reaction to occur though, okay, right? So if I want to do this and I want this to actually happen, Well, let's talk about that. Because it doesn't naturally want to occur. Actually, none of these things really want to happen because I have to get over that activation energy. So let's talk about how do I get energy to actually happen? How do I get a non-spontaneous reaction to occur? Well, that's a great question. So what we're going to do is we're going to couple that reaction with ATP typically, or some other reaction that is exergonic, okay? So if I have a non-spontaneous reaction, which by definition is endergonic, it has a positive delta G, what we're going to do is we're going to either push or pull the reaction through the entire process, okay? So yes, we can look at, and when we look at glycolysis, we're going to see this a lot. You can zoom in on one specific reaction and see that it has a positive delta G. It is endergonic. However, overall, the entire process has a negative delta G because we can kind of push and pull it using ATP, which has a very powerful exergonic or negative delta G. And we can cause that entire process to happen, okay? So in this case, thank you Khan Academy again for a nice diagram. So we've got A, B, C, D. Kind of imagine this is the process of glycolysis. So we're gonna have our glucose molecule come in. So I'm gonna push that glucose molecule in here, and then I'm gonna change it from like glucose to glucose 6-phosphate. Okay, awesome. So that doesn't naturally want to happen, but if I have an equilibrium out of whack, okay, which biological systems never want to have equilibrium. Okay? Equilibrium being equal on both sides, right? You're always heading towards equilibrium, but as a biological system, I want to avoid that. You never want to reach equilibrium if you're a biological system. And so in this case, we're going to use ATP, right? Which is really, really easy to break that phosphate off. It gives me a ton of energy to use. And I'm going to use that with this little pump here and pump glucose in, okay? So that process is going to build up a ton of glucose here, product A, or reactant A. Now, going from A to B may not be favorable, but if I have a ton of A and hardly any B, then this reaction is going to happen. It's going to push it into the reactant here, or it will push it to become this product, I'm sorry. And then we can do the same. So it's going to, as long as we're pulling these things away from the, we're pulling our products away from the reactants, okay? It's gonna pull it through, all right? So we can push it this way, or we can pull away the end product, okay? So in this case, we get all the way to the end of glycolysis, we got our lactate left, and we're going to pull that away, okay? Because we're just trying to break that equilibrium. So the C and the D, if they're in equilibrium, it's not gonna happen. But if I pull all the D away, then I can cause C to convert to D, especially if I use enzymes to do it, okay? So that's how we're going to couple these reactions, which is a term we're gonna see a lot. That's how we're gonna couple these reactions between these things that don't want to happen, that have an endergonic reaction, that have a positive delta G, to ATP, which has a negative delta G, or whatever the negative delta G is. Briefly talk about activation energy. I mentioned that a little bit earlier. So the activation energy is the energy that you need to actually force your molecules to enter their transition state, okay? So it's the energy that you need to get the ball rolling. And we're gonna see that a lot. And when we talk about enzymes, we're gonna talk about how enzymes actually lower the activation energy, okay? So enzymes act a lot like, let's say a common analogy for activation energy would be like you're laying in bed, and you need to push those covers off and actually get out of bed. And once you're out of bed, awesome, you're up, you're doing stuff, you're getting things done, okay? But to actually get out of bed, it takes a little bit of effort. You gotta push those covers off, right? That's your activation energy, the amount of energy that's required to put in to actually get you out of bed. Now, an enzyme would be, let's say you had 20 different sheets and comforters and blankets on you because it's really hot out, or hot out, what? It's really cold out. And so your mom comes along and she says, it's gonna take a ton of energy to get you out of bed. So what if I pull off all those covers, and then it's way easier. All you have to do is just kind of turn and roll out of bed, okay? It takes a lot less energy for you to get out of bed. Well, that's what enzymes do. They lower the activation energy. Now, another example would be, so your mom had to put a little bit of energy into the system to actually get it to happen, right? Another example would be the activation energy needed to initiate a nuclear explosion. And so nuclear weapons, they actually require conventional explosives. So that's what's in the yellow here. And so there's the gun type and the implosion assembly. In case you didn't know this, you'll have like a core of U-235, and there'll be one little, like a bullet size chunk missing, and you're going to use a regular gun basically to, I mean, it's bigger than that, but a regular gun to shoot that little bullet of U-235 into the rest of the U-235, and that energy that you input, that activation energy, allows you to get a ton of extra energy out of the system, okay? Not that we have to talk anymore about how nuclear weapons actually work. This one's similar. You just completely surround it with regular explosives and it compresses the plutonium. But hey, we don't really need to talk about that. But it's the activation energy. I have to put a little bit of extra energy in to get a ton out. That's the whole point. The idea in a biological system is actually going from your reactants to your products. Okay, so in this case, I'm going to have a negative delta G, right? So my reactant energy versus my product energy is different. Okay, so this is a negative delta G, so this would be an exergonic reaction. If my products were up here, it would be an endergonic reaction, and I would have to put more energy into the system, but I'm still going to have to overcome that activation energy. What enzymes do is they make it easier to enter the transition state between the reactants and the products. And how they do that usually is by stabilization. Okay, that's going to be the main way that they do that. And so you can see here when I have my normal activation energy versus with an enzyme, and I'm going to lower that activation energy. Okay. Now, to actually get over this hump, that's where I'm going to have to couple these things with like ATP hydrolysis. Okay, but I can still make it a lot easier. Okay. Now, let's get to talking about an example. Okay, so for example, we're going to see this later on because we're going to spend an entire lecture on glycolysis, and you have to memorize it. So glycolysis, the first step is going from a glucose molecule to a glucose 6 phosphate. Okay, so we take a phosphate molecule and we stick it onto the number 6 carbon. Why do we do that? That is a great question that I like to ask on exams. So it turns out glucose can pass through the phospholipid bilayer. Now, if you have insulin and you get a glucose pump, so that's what insulin does in cells. It causes a pump to be made that can pull in a ton of glucose. But glucose can pass freely through the phospholipid bilayer. Okay, when you have a pump, you can just get more of it. And so the problem with that is it can enter and it can also exit, right? And so we don't want equilibrium. We want, once we get our glucose into the cell, we actually want it to stay in there. And so if we add a charge to that, that does two things. If we add a nice giant negative charge with a phosphate onto our glucose, that's going to attract it to the philic heads, right? The phospholipid heads of our phospholipid bilayer, and it's going to cause it to be repelled by the hydrophobic center of your phospholipid bilayer. Okay? So by adding a charge to your glucose molecule, you can cause it to stay within the cell. Okay? That's really, really important. It's one way that we defeat equilibrium. Okay? Now, we're going to go from glucose to glucose 6 phosphate. That is an unfavorable reaction. Okay? So think back here too, if we're kind of talking about this step here. So going from glucose to glucose 6 phosphate. So to actually do that, that is an endergonic reaction. It's positive. I'm just going to put a plus sign here. It's a positive delta G. Okay, so it doesn't normally want to happen. However, if we look at ATP, which these numbers sometimes vary, I realize that, but we're just going to stick with this one. So if I take my ATP and I break it into ADP and an inorganic phosphate, that is an exergonic reaction. So if I could couple these together, combined, I have a negative delta G, right? Because this negative delta G is so big, it sort of overwhelms this positive delta G. And that's what's kind of happening when we're pushing and pulling these reactions. In this case as well, we're going to take that phosphate that's left over and attach it to the glucose molecule. Whet your appetite a little bit, I wanted to show you kind of exactly what's going on, okay? So we've got our glucose over here. We got our carbons, 1, 2, 3, 4, 5, 6. I counted too fast. And then we've got our adenine here with our triple phosphate chain. So this is adenosine. I guess it's an adenosine. Sorry, it's not adenine. There's adenine. So if it's adenosine triphosphate, and what we're going to do is we're going to use an enzyme called hexokinase to lower the activation energy. Okay, so what we're going to do is we're going to have a nucleophilic attack. Yes, organic chemistry is coming back to get you. I'm sorry. I promise I won't say nucleophile too much, but what we're going to do is we're going to have a nucleophilic attack from these two electrons, okay, on this oxygen, right? Because we have this oxygen sticking off here. It's got two bonds. It's got a bond to this carbon and a bond to this hydrogen, and it's got two extra pairs of electrons. And we're going to have a nucleophilic attack, right? So these electrons are negative. They want to attack something positive. This phosphate is positive. So they're going to have a nucleophilic attack on this phosphate, okay? They want to form a new bond. The problem is there's a ton of negative charge here, okay? So the activation energy for this is a big ask, okay? So it's a big hump to get over. So what we're going to do is we're going to use hexokinase, okay? And hexokinase is going to come along, and hexokinase within its active site, and when we talk about enzymes, we're going to actually talk about the structure of enzymes and what they look like. And within the active site, which is where the enzymes do their thing, it's going to have magnesium ions, which is really, really common. We're going to see that a lot. And it kind of makes sense. If you want to get some electrons doing their thing, if I want to make new bonds, I need to sort of distract the electrons that are around here and kind of pull some of that negative charge that's attached to this positive charge, right? Because the phosphate is positive. It's a positive atom with a bunch of negative charges attracted to it. So I need to pull those negative charges away. So I'm going to attract them to a different positive charge to pull the negative charge away, okay? So that's what hexokinase is going to do. We've got our nice magnesium ions here, and they're going to keep all these extra electrons busy, okay? And so as that does that, that allows our electrons to form a new bond over here. We're going to lose that hydrogen. Okay, we're going to lose that dude just to the aqueous solution. It's going to mix back in with the water molecules, which the next slide set, the next lecture that I'm going to give is talking about buffers and aqueous solutions and things like that. So check that out. But we're going to form a new bond here with these electrons and this oxygen to this phosphate. And so this is our transition state. So now we're kind of in between the two. And the enzyme, our hexokinase, is just going to make within that active site that nice little safe spot for our transition state to safely exist. Okay, so look at that. Look at that animation power. Wow. So we're kind of going between these two things, and then we're going to end up with, I just grabbed, I realize these look a little different. I just grabbed the actual official diagram of what glucose 6-phosphate and ADP look like. But that's what we're left with, okay? So we're left with, we went from two things, 1, 2, to three things, 1, 2, 3, and we now have a glucose 6-phosphate. Awesome. So this whole process is really, really cool, and it only happened because not only do I couple this reaction, okay, with the push and pull of driving glucose into the cell, and then pulling the byproducts away, but we're also going to seal it into the cell by using this process, okay? And even though going from glucose to glucose 6-phosphate has a positive delta G, okay, so that's this molecule going to this molecule is an unfavorable reaction. However, we are coupling it with ATP hydrolysis. And that is the key, and that is why we can do thermodynamics and biochemistry. Just a few points to sum things up. Remember, just going to go over this one more time. The big points. Entropy is chaos. So if you are increasing your chaos, then that is a positive delta S. If you are decreasing your chaos, that means you are becoming more ordered. Enthalpy is bond energy. So if you break a bond, you're releasing energy. That's a negative delta H. Energy is coming out. If you are building bonds, that is a positive delta H, okay? So your enthalpy is going up, okay? So this would be exothermic, and this is endothermic. Gibbs free energy is just a simple way to tell if a reaction is going to be spontaneous, okay? It's us combining temperature, chaos, and enthalpy to make one number that we can then use. Now your delta G, if it's negative, is spontaneous, okay? That doesn't mean it's instantaneous, but it is spontaneous. That means it can happen. Basically, we're just comparing reactants to products, okay? So is it going to happen spontaneously or not? Or do I have to put a little extra energy in, okay? If it's a positive delta G, it's non-spontaneous, so I need to put extra energy into the system. It doesn't mean I can't do it. It just means it doesn't happen, or it doesn't want to, okay? But I have to put extra energy in to get it to happen. You can overcome these non-spontaneous reactions by coupling them with spontaneous ones and by applying pressure from reactants or removing products, okay? So if I put a bunch of reactants in, or I pull a bunch of products out, That's how I can push and pull these entire processes, okay? Lastly, we can use enzymes to actually lower the activation energy of reactions. And because they stabilize the transition state of molecules, that's how we're basically going to do it. That's why we make so many enzymes in our bodies, okay? Hopefully that is helpful. Stay tuned for more about enzymes and how enzymes actually work, and we're going to go through some very specific examples, and also check out the next set of slides, because that is going to be talking about pH and pKa and protein pKas, all kinds of fun stuff.